Greetings, AP Bio students. Welcome to your fifth Prezi for evolution. This is classification. Before we get started, I do just want to give a big overview for this because there aren't a ton of notes to write for this particular Prezi. What's most important for your understanding of this material is actively practicing these cladistics, actively practicing making cladograms and interpreting that data. So whatever time you get to save this evening from your homework of taking notes, please make sure that you're doing those mastering problems of creating phylogenetic trees. You will not regret spending your time in that way. So just quickly, I want to review some of this material with you. So old school versus new school versus the actual application or the cladistics piece, which is the more challenging part to understand. If you remember doing your critter project freshman year, many of you would have organized your organisms using the old school kind of terminology. While some of that is still applicable today, the vast majority of what we use in biology is now based on genetics. So this is going to be the more important information of these two. Okay, so is this a snake or is it a lizard? Big questions for this presentation. What is the pattern of life's diversity that has developed during the history of life on Earth? How do we determine evolutionary relationships between organisms? And what are the characteristics of life that are useful for determining evolutionary relationships? In the 1700s, Carl Linnaeus came up with two big contributions. The first is that he decided to classify species based on similarities, and the second is he developed the binomial nomenclature that we still use today, and that's that species scientific names have two parts. So there's the genus and the species. You need to make sure that you always capitalize the genus and that the species is always lowercase. So an example of this would be Homo sapiens, and that's for human. That's the scientific name for humans, or the using binomial nomenclature. We're going to still use this in class, so it's important that you kind of get used to recognizing that. The hierarchy of life, and moving from domain to species, the number of possible groups increases. So if you look at the very bottom, there are three different domains. So we've got bacteria, eukarya, and archaea. From that, those three things are based on cell anatomy. And then from that, there are five traditional kingdoms. So we've got Monera, which is bacteria, Protista, which are protists, fungi, plantae, which are plants, and Animaliae, based on cell anatomy and nutritional modes. But these are not scientifically valid anymore. So there are about 2 million described species, and the total estimated of all species is between 10 and 100 million of species. If you look at the three large domains, we've got bacteria, archaea, and then eukarya. You're probably most familiar with eukarya because it includes animals and plants and fungus and protists. Okay, so it turns out there were some issues with the old style, and so we've now included some ways to more formally classify organismal relationships. So first thing that you should know is that the number of kingdoms, it kept changing. So first there were 2 kingdoms, then there were 3, then there were 5, then there were 6, and there were 8. A lot of the early work was based on visual observations. That was the second problem. So looks can actually be deceiving. Why do you think that might be? The second issue, or related to that, is that described species are heavily macroscopic, and that's just because we can see them. And it turns out that almost all of the species on the Earth are microscopic. So it's curious that we have an organizational system based on what we can see, whereas most organisms on the Earth are small that we can't see. And so if you thought about this first bullet point, looks can be deceiving and why, look at these two creatures, you might think that they are related to each other, pretty closely related, and you might even be tempted to put them in the same classification group. But you'd be mistaken to put them pretty close together because this top organism here is a marsupial mole, whereas the bottom is a placental mole. So their reproductive strategies are different and their development are different. And so the reason they have similar kind of body plans is because their ancestors evolved under similar pressures, right? So these are examples of analogous structures rather than homologous structures. And so that is one of the problems with the kind of old school way of classifying organisms. So whenever we discovered DNA and started to organize creatures based on their DNA, it really helped understand relatedness among organisms. It is a reliable indicator of relatedness because as species diverge from one another, their DNA sequences diverge as well. And so this has revolutionized classification. You take a look on the right, you can see examples of mutations on two homologous DNA sequences. And so what we see is we've got a deletion, which means that one of the nucleotides was taken out accidentally. And then we also have an insertion down here. So an insertion is just the addition of a few nucleotides. And so what that results in then is two different DNA sequences that are still reasonably similar to each other, with the exception of that deletion and the insertion. And so what we do then is we can actually track those changes. And so the way that it works is that we determine evolutionary relationships among organisms using the overall rate of mutations in genes to act as a molecular clock for dating evolution. So there are different genes and different organisms have different mutation rates. But if you look here, we see the divergence time in terms of millions of years on the x-axis, and on the y-axis we have the number of mutations. And if you draw a best fit line throughout the data, you see that as time goes on, so divergence time goes on, so millions of years, we see the number of mutations pretty much steadily increases. And so we can use that to track how similar or how dissimilar. One thing that you should note is that gene duplications play a large role in evolution. And so what happens sometimes is you might accidentally get a gene duplication in an organism's DNA. We'll go over how those mistakes might happen whenever we discuss DNA replication. But just for now, know that if you've got extra DNA floating around, or in the chromosomes rather, but still have a functioning gene somewhere else, it kind of allows mutations to accumulate in the second set of genes without affecting the function of the original gene itself. And so we'll discuss more about this whenever we actually discuss protein production and DNA replication, but know that I do need you to know that gene duplications play a large role in evolution. Okay, so DNA evidence has also identified endosymbiotic ancestors. Again, this is something that we'll touch more on later, but it's helpful to recognize that DNA has really clarified organismal relationships. So much so, even between relationships between organisms far back as 2 billions of years ago, we can use this evidence. I'm here to pause this video to review the current phylogeny of life. Okay, so into cladistics. So first of all, you should know that this is how we group organisms according to the number of shared characteristics they have in common. And this is what we refer to as a cladogram. It can be based on any characteristics, but ideally it is based on what we call shared derived characteristics from a common ancestor. All right, so to make a cladogram, you first have to figure out how many species have a particular characteristic in common. Then you have to group the species so that the most number of species have the most characteristics in common. I don't know how helpful it would be for you to actually write those things in your notes, so instead we're just going to look at an example together here. So if you look at this chart here, okay, this character table, if you will, the way that you read that is by identifying which organisms have which characteristics in common. So the vertebrae column, or backbone, you can see that this organism, the lancelet, is the outgroup and it does not have a backbone, whereas the lamprey, the bass, the frog, the turtle, and the leopard, they all have a backbone. Hinged jaws is another shared derived characteristic. You can see that this organism, the lancelet and the lamprey, they do not have hinged jaws, but these organisms do all have hinged jaws. And you can do the same thing with the four walking legs, the amnion, and the hare. And so what you can see here is that the leopard has all of these traits, but it shares a lot of traits with the other organisms in the line here. And so if you look at the tree that we've created, is you can actually draw these shared derived characteristics onto this phylogenetic tree. And so what we see here is that it is good to have an outgroup. So this is an organism that's really farly distantly related to organisms. This node here, and it would be helpful to sketch out in your notes what a phylogenetic tree looks like. This node here is going to represent a common ancestor that existed and a speciation event. So common ancestor and a speciation event, or a shared common ancestor, excuse me. Then here we have the lamprey. You can see that the lamprey, before it deviated from its ancestors on this side of the tree, the common ancestor here had a vertebrae column, which means that all of these organisms from the lamprey to the leopard have a backbone. And so just like that, we can kind of see which organisms have this hinged jaw. So if I were to say which organisms, according to the phylogenetic tree, have four walking legs, you would go down to this shared derived characteristic. And then any creatures that come after this would share that particular trait. So the frog, the turtle, and the leopard, their ancestor all had four walking legs. Also important to note that this axis, though it's not labeled, does typically represent time. So more recently, the turtle and the leopard shared a common ancestor than say did the bass and the leopard. Let's see. The bass and the leopard, they last shared a common ancestor all the way back here, right after the hinged jaws. So the bass and the leopard shared this common ancestor. So we would say the bass and the leopard are not as closely related as the turtle and the leopard, because the turtle and the leopard more recently shared a common ancestor. It's important that you understand how to read these. So any questions at all that you have, I'm going to need you to pause the video and write them down now so you can bring them to class. The more specific questions that you have, the better. The second piece that you might have a kind of question about is, you might think, oh, well, aren't all of these like characteristics, aren't they kind of macroscopic observations? And while yes, they are, they're also confirmed in the organism's DNA. So that's important to know, that these shared derived characteristics are based in similar DNA sequences. Okay, so the last piece of this is that you should know that the rule of maximum parsimony applies. So what this means is when you're creating these trees, all other things being equal, the simplest explanation is true. So characteristics are most likely to evolve once. So the rule of maximum likelihood says that assuming that mutation rates are equal in different lineages, more closely related organisms will have fewer differences in genetic sequences. And so what I mean by that is, if you look up here back to our phylogenetic tree, yes, it's possible that the hinged jaws evolved like separately in the bass's ancestor, only the frog's ancestor, only the turtle's ancestor, only the leopard's ancestor. But more than likely, that probably only evolved once in this lineage. So that's what the rule of maximum parsimony is trying to tell us. Okay, so here we have three different species. Species 1, 2, and 3 of these different bird species. And so which tree is the best? Because we could organize them so that 1 and 2 are most closely related, or 1 and 3, or 3 and 2. And so the technique involves figuring out the shared bases. So we can see in my ancestral sequence, So this is kind of like the outgroup, if you will, or an ancestor to all three of these species. We see it has a sequence of A, G, T, T. So looking at these species, we would say, okay, this has the same ancestral sequence. It also has an A and its first site, as well as the second site. It also has a G, just like the ancestral sequence. But then we can see here that we have an A where there used to be a T, and a C where there used to be a T. And then so this species 3 has 2 differences between the ancestral sequence. Species 2, let's see, it's got 1 difference, 2 difference, 3 differences, and then species 1 has 1 difference, 2 difference, 3 differences as well. And then you could start putting them on a tree, and unfortunately what you get whenever you apply that maximum parsimony is you get a whole bunch of different options, and this takes a really long time to do. So seriously, we will just use a computer to make these things happen, and we're not going to make your brains hurt like that, if you will. But do pause this video for a second and follow what these changes would look like. Okay, so there are 3 different types of groups, and I don't need you to memorize these definitions. What I need you to understand is the application. So before you write anything down, listen to what I'm describing here. So monophyletic groups are pictured here. These are all descendants of one common ancestor. So notice how A, B, and C all have a shared common ancestor here. So we would call this a monophyletic group. A paraphyletic group is some of the descendants of one common ancestor, just like this. So some of the descendants, but see how we neglected G? So it's not monophyletic because it only has some of the descendants of that one common ancestor, which is here. And then finally, the polyphyletic group. These are the descendants of more than one common ancestor. So here we've included A, B, C. We also have D, whose common ancestor is not until we get down here with A, B, and C. And so what's important to note is that a major goal of phylogeny is to develop a cladistic system that consists of nested monophyletic groups. So these are going to be the groups that we look at and that we compare and that we care about. We're typically not going to be drawing conclusions and trying to organize trees in this way. Ideally, whenever we're talking about a group of organisms, they're all leaving out some of the descendants of a shared common ancestor. Again, if you have questions, write them down right now. Don't stay confused. Bring them to class. Example of a phylogenetic tree that correlates DNA changes to geological time. So here we've got present day time, and then on the x-axis we've got millions of years ago. So this is just organized just based on time periods. And we can see that a human and a mouse most recently shared a common ancestor 65.5 millions of years ago. So we would say that these 2 organisms are more closely related than, let's say, a chicken and a human, because a chicken and a human haven't shared a common ancestor since more than 251 million of years ago. Okay, and so this is an example of a phylogenetic tree that you should be able to read and interpret. All right, any questions? Besides, of course, that this is a lizard and not a snake. Please do make sure you can do a couple of things whenever you're done. So first of all, explain how hierarchical classification of life and binomial nomenclature work and how they demonstrate evolutionary relationships. Tell me why classical approaches to classification have needed to be overhauled recently due to advances in molecular biology. Be able to explain the process of creating a cladogram and demonstrate that the process, if given a list of shared derived characteristics for a group of organisms, by applying the rules of maximum parsimony and/or maximum likelihood. Be able to identify a taxonomic group as mono, para, or polyphyletic, and explain the relationship between classification and phylogeny.